Alright, the last part on the recombinant gene expression is actually on gene editing. So gene editing is basically a normal occurrence or normal mechanism and is a part of everyday's life, but it could also be inherently dangerous. And there are various forms of gene editing, and this is done both in prokaryote and especially in eukaryote. So the forms of gene editing are organisms' own gene editing during transcription and translation. So no organism is an exception to this. And there are also assisted or manipulated gene editing such as knockout, knockdown, knock-in, and also CRISPR. So we're going to take a look at this four and how it could be used nowadays in this modern world. So the differences between knock-in, knock-out, and also knockdown. So I'm not sure whether you have heard of all three or at least you have heard at least one knockout. So knockout is the most common, and for I think some of you here have taken my class in during your undergrad, so you probably would know this. So knockout is the most common of gene editing. Okay, this is to replace or disrupt anything of the gene with the coding exons with a selectable drug selection marker. So what you could do is that you make sure that the gene doesn't function. You knock it out. So you knock out the function, and the rationale behind gene knockout is basically just to know how the organism would behave or whether it could survive without having the gene expressed. Now, knock-in, you can basically read all about it. Basically there are lots of information down here, but I would just simplify it in this video is that for knock-in, so usually what happens is that the DNA sequence is being mutated, and or it is exchanged or swapped for the sequence which is not disrupted. So you usually just swap the gene function in terms of the knock-in, and you wanted to know whether a certain part of the gene when it is being swapped the sequences, could it still function, could it not function, and what would happen to the system or to the organism overall, or just in certain organelle. And the last one is the knockdown. So for the knockdown, usually technology used is the RNA or RNA interference. So simply said, genes are silenced or knocked down by short pieces of double-stranded RNA. Simply said that this is different from knockout. Knockout, which means that the gene is being basically kind of like turned off. It will not be turned back on. But in knockdown, via RNA, the genes are only silenced for a certain period of time, or if you want to silence it for the whole duration, it's up to you. But the gene could still be turned back on. It's just that it is being silenced. So the function is just to see how the organism or the system will behave when the gene is being silenced. Could it survive? Because in certain study, RNA is actually quite useful, especially when a certain gene, when a certain protein is being produced too much or in abundance. So RNA is usually being used to control the gene expression. Now, so these are the differences between the three. You can have a look in your notes of the side-by-side comparison, and basically basically this would be the definition of how basically it could be done, and how it is being identified. So complete loss of genotype, gene itself is still intact, it's just that it's not expressed, and not gain of function or loss of function of mutation. So, and in terms of how long does it take to do it, and also whether it's going to be a lethal conditional knockout or not. And some of them, such as knockdown and knock-in, they have some extra feature. So for example, for knock-in, there are difficulty levels and time taken vary on gene's choice, because when you basically want to do a swapping, and especially when the gene comes from two different organisms, let's say prokaryote and eukaryote, so you will need a lot more extra time. So for knockdown, there are two extra features, relatively easy and fast to perform because you are just silencing it and just by using RNA. And also it is possible to carry out embryonic lethal gene to behave similarly to knockout, just that the gene is not being shut down completely, just silenced. Alright, so we move on to the last part which is on CRISPR. So I'm hopeful that at least some of you would have heard about CRISPR, not the crisps that you eat. Well, in UK they call it crisps, over here we call it chips, right? So CRISPR is for clustered, regularly interspaced short palindromic repeats. So if you remember your palindrome, this is basically that kind of palindromic repeats. So it's first reported in 2012 by Gene et al. And first journal coming out from 2012 as well. So what happened is that basically this was done in Drosophila, and Drosophila, okay, as we know, okay, is a very small animal. Okay, so it silenced foreign nucleic acid, okay, adaptive immunity against virus and plasmid. So the mechanism used is just Cas9 enzyme, okay, with a guide RNA molecule to surround the target DNA for the editing. So basically, okay, you just have a Cas protein with another guide RNA go to the site, okay, to be edited. Okay, you usually just cut it out, okay, and then replace it, okay, with the gene that you want to be there, okay, ligate it, okay, and then work done. So it's basically has been reported to be cheap and fast, and has been proven to also be cheap and fast, easy to use, and so far any organism's DNA can be quickly changed. But basically in human or in mammalian, that still needs a lot of investigation, because thus far not many report basically could confirm, okay, that it is fast. Cheap, probably. Fast, nobody would want to ascertain that. Now, basically, CRISPR is adapted from bacteria, and of course it's by utilizing the bacterial resistance against viruses. So this is done to accommodate precise insertion of the DNA into the genome of any organism. So CRISPR sometimes could cause unwanted changes to the genome, and this is basically something that is unwanted, or something, okay, that you couldn't do without it. Sometimes, okay, what happens is that you could activate genes, okay, that cause certain cancer and so forth. So you need to be very careful when you basically use CRISPR, or when you use gene editing via CRISPR. So how do CRISPR works? So it's actually fairly simple, okay? So like I said, you need a Cas protein, and in this case, the first protein ever used is Cas9. I think previously, okay, from 2018 onwards, they reported that there are 10 Cas protein that could be used for CRISPR. Cas9 would be the famous one, but I think as of earlier this year, there are about 15, 20, or probably more of the Cas protein that has been identified, but have to be tested. Okay, so Cas9 protein, okay, have to form a complex, okay, with a guide RNA. So this is your Cas protein that looks like a cloud. The guide RNA, okay, is this one, the line here. So guide RNA is actually very crucial apart from the Cas9 protein. So when this form a complex, okay, it will attach to a matching genomic DNA sequence which is adjacent, okay, to a spacer, okay, whereby you have this. So this is where, okay, once it attaches itself to there, okay, the Cas9 RNA complex will cut the double stranded of the DNA, okay, at the site, and the program, okay, DNA may be inserted at the cut to replace the one that has been cut off, okay, and how it is being incorporated or inserted into by using ligase. All right, so enhanced, okay, you can get it done, okay, with a short period of time, and it is actually quite fast and cheap. So by utilizing CRISPR, okay, the same mechanism of gene activation and also gene repression could also be done. So we have studied this a lot previously, okay, for the past few weeks, okay, on how gene could be activated, or how it could be expressed by using the operon system, especially in prokaryote. So in terms of using CRISPR, this could also be done, whereby, okay, when you have your Cas9 along with your guide RNA here, okay, whereby when you have an activator and you want the gene to be basically incorporated and expressed, okay, so this could be done. And when you want to have it repressed, okay, you could also do the same thing, and this has been found, okay, to basically reduce the time to activate and also repress gene, okay, and at a lower cost as well. Apart from that, okay, another thing that usually, okay, a lot of researchers, especially when they are doing a research at university or research institution, okay, is that they wanted, okay, to know whether the gene could be expressed, or how long it is going to be expressed, okay, or how much it is being expressed, usually by visualization. So using similar CRISPR approach, okay, but okay, in this case, okay, you basically use a GFP, or green fluorescent protein, that will fluoresce under basically UV excitation, okay, to see the expression of the gene, okay, whether you have it edited or not. So all in all, to sum up, okay, on the CRISPR, is that, okay, it is fairly easy to use, and like I said, okay, it basically takes a model, okay, from bacteria, and this is basically how the idea came. Okay, so the first stage is actually for the foreign DNA acquisition, okay, whereby okay, Cas9 gene okay, is being released okay, together with your guide RNA. So over here probably you don't see okay, the guide RNA. So second stage okay, is that the CRISPR locus transcription occurring okay, whereby you get CRISPR RNA processing. Okay, over here you get your pre-CRISPR RNA being made. Okay, so your guide okay, RNA okay, will be the ones okay, that will carry or basically form a complex okay, along with your Cas genes okay, and this is usually packaged okay, and for example this is done again using viruses, packaged and to be used again on other organisms for gene editing as well, and this has been commercialized for use, so you can actually buy a CRISPR kit nowadays. Of course it costs about quite expensive, but it's actually fairly easy to use. Right, so there you have it, the gene expression system. If you have any questions, so like I said, okay, we will have a discussion later on Monday.